Hello my dear friends, this is Dr. Pooja, your biochemistry faculty. Well, in this video we are going to discuss about vitamin B6. Now we all know that if we talk about vitamin B6, it is a water-soluble vitamin first of all, right? And B6, that means it belongs to B complex. That means I have told you one very important thing about the B complex vitamins, that they are used as coenzymes. Whether we talk about B1, whether we talk about B2, B3, B5, all of them are working as coenzymes, means they are acting with the enzymes in the so many biochemical reactions. So acts as a coenzyme. Now if we talk about the active form of vitamin B6, so it is actually known as pyridoxamine, pyridoxal phosphate. So I'll just show you the structure of this. So it can exist in these forms. This is the basic structure as you can see, but the only change would be in the R group. Okay, so this R group, if it is replaced by CHO, it becomes pyridoxal. And the active form of pyridoxine, or you can say B6, is pyridoxal phosphate, or commonly we call it as PLP. Right, very, very important. So I can say that the active form of B6 is PLP, which stands for pyridoxal phosphate. Now if we talk about the sources here, sources of most of the B complex vitamins are more or less the same, like meat is there, egg is there, milk is there. They are the common sources. You can say green leafy vegetables, right? So there is no specific source for PLP. But yes, if we talk about the functions are this PLP works as a coenzyme for variety of biochemical reactions. So what are the biochemical functions? The most important function of this PLP is it is a coenzyme for transamination. So I can say the first reaction which is being catalyzed by a B6 coenzyme is transamination. Now what is transamination? We all know transamination is simple transfer of an amino group. From where? From an amino acid to a keto acid to form a new amino acid and a new keto acid. So be it alanine, be it aspartate, they both will undergo transamination, right? And the reaction will be the same. 𝗵𝗵𝗺𝗺𝗺, here will be the keto acid, 𝗵𝗵𝗺𝗺, here will be the keto acid, forming glutamate plus pyruvate, which is a keto acid derived from alanine. Similarly, here glutamate will be there and oxaloacetate will be there. And we all know that this is catalyzed by the reaction by the enzyme ALT, and clinically it is known as SGPT, whereas this is AST, or it is known as SGOT. Both are diagnostically very, very important enzymes. If there is anything wrong with the liver, GPT will always show arise. If there is anything wrong, just say if a person has MI or any liver disease, then it will be GOT. So it is a cardiac marker as well as liver marker. Similarly, if we talk about GPT, it is raised in non-alcoholic fatty liver diseases. And SGOT raises in liver neoplasia, or you can say cirrhosis of the liver. So these two are very, very important enzymes. Coming over to PLP. PLP is always used as a coenzyme for all the transamination reactions in the body. So I can say transamination ke liye the coenzyme is always PLP. Similarly, one more reaction is there in which glycine is involved. So if we talk about glycine, again glyoxylate is there. right? And this glyoxylate can be converted into oxalate if PLP is absent. And this oxalate will lead to the precipitation of the stones leading to oxaluria. So, transamination always requires PLP as one of the major coenzymes. Second reaction, if we talk about, the second reaction is transsulfuration. Again, it is clear from the word itself, transsulfuration means there is a transfer of sulfur, or it is related to sulfur containing amino acids. So, in transsulfuration, cysteine is produced. And how cysteine is produced? Simple reaction is there. This homocysteine, which is obviously derived from methionine after donating a methyl group, this homocysteine is first of all converted into a complex which is cystathionine. And if we talk about here, serine comes. And the enzyme here is cystathionine beta synthase. This is the name of the enzyme, and again PLP or B6 is used as a coenzyme, and this cystathionine splits up. The splitting enzyme is there and cysteine is produced, giving out homoserine. So I can say B6 is required for the synthesis of cysteine, and cysteine is again further a glucogenic amino acid because cysteine is responsible for the formation of cysteine, and it is a glucogenic amino acid. It forms glucose in the body. So B6 is required for transsulfuration. Then third important process is decarboxylation. Now, if we talk about decarboxylation, simple removal of carbon dioxide is there. And if we talk about the major reactions of decarboxylation, the most important which you all are familiar with is histidine. This histidine will undergo decarboxylation to form histamine. Histamine, which is released by the mast cells of the liver. And this histamine is actually raised in the allergic reactions in type 4 hypersensitivity. So, histamine, which is actually produced as a result of B6. Similarly, if we talk about other products, tryptophan. This tryptophan undergoes hydroxylation first and then decarboxylation, forming 5-hydroxyltryptamine. What is this 5-hydroxyltryptamine? It is none other than your serotonin. And we all know that serotonin is a very, very important vasoconstrictor, and it is always released in the body only when, you can say, when somebody is like in a depressive mood. So it elevates our behavior. It elevates the cerebral activity. And then further it is degraded into 5-HIAA. And the synthesis of serotonin from tryptophan I have already discussed in the video on the metabolism of tryptophan. So I can say for serotonin also B6 is required. So wherever you will find a decarboxylation, always keep one thing in mind, the coenzyme required is B6. And there is a difference between decarboxylation and oxidative decarboxylation. Whenever this removal of carbon dioxide is taking place in the presence of oxygen, it becomes oxidative decarboxylation, which is there. For that the coenzyme is B1 or TPP. But whenever simple carbon dioxide is being removed, it is B6 always. So these are the two. In fact, there are so many reactions related to this, but we are just discussing these two. Similarly, tyrosine can also be converted into tyramine, right? Dopamine, so many are synthesized in the presence of this B6. Then I can write one more reaction here, that like tyrosine forming dopamine. Again, B6 is the one which is required here. So, for decarboxylation again we say it is required. Then the fourth reaction is that this is a very, very important component of glycogen phosphorylase, very important. So, I can say fourth function is that component of glycogen phosphorylase. Now, what is this glycogen phosphorylase? This is the rate limiting enzyme of glycogenolysis. So, this B6 can also be used for the breakdown of glycogen to release energy, obviously. So, again this is a very important coenzyme. Then another function of this is that PLP is a component of ALA synthase. Component of ALA synthase. Now, what is this ALA synthase? This is the first rate limiting enzyme of heme synthesis. The first rate limiting enzyme of heme synthesis is synthesis of aminolevulinic acid, and this takes place in the mitochondria. Very important, because we know that heme synthesis takes place in two organelles, mitochondria, cytoplasm, means it starts from the mitochondria, goes to cytoplasm and terminates also in the mitochondria. So, if we talk about the first reaction, it requires PLP, and the reaction is glycine combines with succinyl coenzyme A, right? And here again the coenzyme is B6 and the enzyme is ALA synthase, and it forms what? It forms aminolevulinic acid. Aminolevulinic. Right. So, these are the important biochemical functions of B6 that it is required. First of all, if we talk about the first reaction is a very, very important reaction, transamination. Every protein, every amino acid, whatever proteins we are taking in our diet, they will always be degraded as per their carbon skeleton to form urea in the body, right? And this transamination is the process which is taken over by all amino acids. I mean, like there are 20 standard amino acids, so 17 amino acids undergo transamination to release that amino group, which is expelled from the body in the form of urea. So, for transamination, PLP is required. Second, if we talk about transsulfuration, this is also a very, very important reaction. And we know homocysteine. This homocysteine is actually nowadays considered to be a marker of MI also, and it is basically converted into methionine. But here what happens is, this homocysteine in the presence of B6 is going to form cysteine, and this cysteine will further form cysteine. Because whenever we talk about biochemistry, there are so many similes there. Cysteine, cysteine. The spelling is different and obviously the pronunciation is also different. Then decarboxylation. Decarboxylation, a removal of carbon dioxide. We can say histamine is produced, serotonin is produced, and dopamine is produced. Again, serotonin, a very, very important neurotransmitter. Dopamine, very important neurotransmitter, synthesized from tyrosine in the body. Because we know if there is decreased synthesis of dopamine, it leads to Parkinson's, which is seen in elderly people, right? Then it is a component of glycogen phosphorylase also. Okay, then it is a component of ALA synthase. This is also a very, very important aspect of B6. When B6 is present, then only I can say heme synthesis will also take place, because this aminolevulinate will further, later on it will form heme. So whenever there is increased production of heme, it starts inhibiting its own synthesis. So this heme is synthesized with the help of vitamin B6. We can see the reaction here. Glycine will combine with succinyl coenzyme A. This reaction takes place in the mitochondria. And we all know that heme is synthesized in the body. Whenever there is increased concentration of heme, it starts inhibiting its own production. And where it inhibits? It inhibits on the rate limiting enzyme, in which the coenzyme is B6. And we all know this also, that this ALA synthase is of two types, ALA synthase 1, ALA synthase 2. Right. Then another important reaction catalyzed by B6 is in the metabolism of tryptophan. So in tryptophan metabolism, what happens is, this is a very important reaction in fact. We know that tryptophan is a heterocyclic amino acid because it has a characteristic indole ring along with that benzene ring. So what happens is, this tryptophan, when it undergoes synthesis of niacin, there is one very interesting step in which this 3-hydroxyl kynurenine is converted into 3-hydroxyl anthranilate. So what happens in this reaction? Later stages mein this is the one which is going to form niacin. So what happens? This enzyme, kynurenase, requires B6 as a coenzyme. Okay, so without B6, niacin synthesis will also be defective. So these are the important biochemical functions of PLP or B6. Suppose if I take the example of this, what will happen if B6 is absent? It will lead to increased concentration of 3-hydroxylkynurenine, and this will be converted into xanthurenic acid. So in a patient, if xanthurenic acid is being excreted in urine, it is an indication of B6 deficiency. Okay. So that we are going to discuss in the deficiency manifestations. So these are just the functions. So functions may, if we have to summarize these functions, ALA synthase, and the last function is it helps in the metabolism of tryptophan, or you can say it helps in the synthesis of niacin. Then we come over to, after these functions, then we come over to the deficiency manifestations. Now, if we talk about the deficiency manifestations, we are going to take up the first reaction, transamination. So if I talk about transamination, I told you that what will happen is that glyoxylate will be converted into oxalate. Okay, and this oxalate will lead to oxaluria. So I can say that this is in B6 deficiency. Why? Because transamination is defective. Then when we talk about the second reaction is that homocysteine is converted into cystathionine, or I will just write two reactions in one go, forming cysteine. So again, if B6 is deficient, there will be increased concentration of homocysteine leading to homocysteinuria. And this homocysteinuria, again supplementation of cysteine will be there, or supplementation of B6 is required. And again homocysteinuria, this is classic homocysteinuria, one is non-classic homocysteinuria, which I have discussed in detail in the video on homocysteinuria or the metabolism of methionine. So here we will be just concentrating on homocysteinuria will be there due to B6 deficiency. Then the third reaction is again you can say tryptophan metabolism. So here I can just write the main thing. In tryptophan metabolism, due to B6 deficiency, we saw that niacin synthesis will be defective, right? And in B6 deficiency, there will be excretion of xanthurenic acid in urine. So, xanthurenic acid appears in the urine of a patient who is B6 deficient. Coming over to the fourth manifestation, fourth is synthesis of heme. So, if we talk about the synthesis of heme, we know that it is a component of this aminolevulinic. So, again, if B6 is deficient, since it is a component of ALA synthase, it leads to anemia. And the anemia caused by this is microcytic anemia. Right? So, these are the deficiency manifestations of vitamin B6, which plays a very, very important role. Again, if we have to summarize, we can say convulsions will be there, neuropathy will be there. Why? Because if we talk about this, GABA will not be produced, histamine will not be produced, because these are all the biogenic amines which are produced as a result of degradation of the respective amino acids. This is serotonin will not be produced because serotonin is again a very important neurotransmitter. Dopamine will not be produced. So, there are so many important biogenic amines which will not be produced in the absence of B6. So, other manifestations can be, we can simply say that convulsions will be there. We can say seizures will be there. And there will be peripheral neuropathy. There will be tingling sensations also. But yes, there is a difference between a patient who is having B6 deficiency or who is having B12 deficiency. Because peripheral neuropathy is there in B12 deficiency also. But again, the reasons there are different. And here the reason is altogether different. Okay, B12 we are going to discuss after this. So, you can say that these are the deficiency manifestations of vitamin B6, which is known as PLP, which is present in three forms: pyridoxal phosphate, pyridoxamine, and simple pyridoxine, you can say. And if we talk about the RDA of this, the RDA can be 1 to 2 milligram in a day. And then if we talk about the biochemical assessment, this is also very important. So, biochemical assessment mein we can say that xanthurenic acid excretion, right? Second is, you can say homocysteine, like this. So, these are the two main tests for the biochemical assessment of vitamin B6. So, this is all about pyridoxal phosphate. I hope you have understood the main thing, that it is used as a coenzyme for so many reactions. So, that's it. Thank you so much.